kidneys. We're going to be studying renal anatomy and physiology, sometimes called the urinary system. So we'll be making a big transition. Just want to point out that the first exam is on Moodle, as are my answers. You should compare your answers with mine and see where you lost points. And if you have questions, feel free to contact me about anything. So today is going to be an atypical Wednesday because there is no problem set due this week. So we're not going to break out into Zoom rooms to work on a problem set because we don't have one. So today is going to be more like a Monday class. There's going to be at least 2 and maybe 2.5 or 3 different classes going on. And so here it is. So what I wanted to point out here is that the last class from the respiratory section is duplicated here into this section to remind you when you're studying for this next exam that this PowerPoint was not, this class was not on the previous exam and will be on the next exam, as will some of the respiratory stuff, because we'll see that the Davenport diagram, when we get to understanding renal compensation, that's what we're going to be talking about now. So here are the key concepts list, and here are the 3 class PowerPoints that we'll be working on today. Maybe we won't get through it all. We'll see. In lab this week, we will be doing some pig kidney dissection. We will be looking at some pictures, microscopic pictures from microscopes that show you the histology, the cell structure of the kidney. So we'll be doing that in lab. And we'll be doing human cadaver work. The problem with the human cadaver work is that the kidneys are behind the digestive tract in a lot of it. They're behind the liver and behind the pancreas and the spleen. So to get to the kidneys, we're going to start to explore the digestive tract first on our way to getting to the kidneys. Leah, can you hear me now? Okay, good. So let's get to work. Yeah, pig kidneys, human histology, and human cadaver dissection. No problem set this week, but there is a quiz this week, which will be available on Friday and due on Sunday. It will cover the material from today. And then just a note about the next exam that's coming up in less than 2 weeks now. Let's move fast. Okay, kidney. Start moving fast. So we're going to start with the anatomy of the kidney. Well, yeah, we're going to no, let me start. So this is just a picture of the kidney. We are going to talk about blood flow to and from the kidney. We will talk about blood flow within the kidney. We'll talk about these structures. We're going to get to this in a minute. Before I get into the anatomy, I want to put this in the context of physiology. So what does the kidney do? What are we going to be talking about? Well, we're going to be talking about acid-base and water balance, because the kidneys are essential for maintaining body water, hydration, as well as working in cooperation with the lungs, maintaining pH. So we're going to get back to acid-base balance. And then the removal of metabolic wastes and other toxins or foreign chemicals in the urine is something that you're probably already thinking of. The main process of the kidney, the sort of the flow chart of operation of the kidney, is to make a filtrate from blood that is very dilute, and it's very large volume. And then it's going to go through a process of reabsorption to reclaim most of the water and salts and glucose that was filtered. So we make a dilute filtrate that's got a lot of good stuff in it, water, glucose, salts, other small molecules, and then we recover most of it. And then we excrete the rest. So just to illustrate the amount, the volumes we're talking about, here is a 5-liter beaker that shows the cardiac output, mils per minute, 5,000 milliliters per minute, a typical cardiac output, the amount of blood your heart pumps out each minute. About 20% of that cardiac output goes to the kidneys. So 1 liter per minute of blood goes to the renal artery. In that 1 liter of blood, there is about 600 milliliters of plasma, of serum, because 40% of the blood is cells, mostly red blood cells, and 60%, 600 mils or so, is plasma. And I'm not going to hold you to those numbers, but you can think of it as 50-50. Half of the blood is cells and half the blood is plasma, although it's more closer to 40-60. The word there is hematocrit. Hematocrit is the official word for the proportion of blood volume that is cells, that's red blood cells. So about 40% of blood is red blood cells, and the other 60% is plasma. Your kidneys filter about 100 mils of plasma every minute. So every minute, your kidneys are taking this much volume out of blood. Okay, so you can imagine that in about 5 minutes, you have about this much filtrate. And so if we weren't reclaiming the water, you'd be spending your whole life in the bathroom, because that's about the size of your bladder, right? I mean, that's about how much urine you void every time you go to the bathroom. So in 5 minutes, in about 20 minutes, you'd be completely dehydrated, because this is how much fluid you filter every minute. And this is about how much fluid you excrete every minute. So the difference between this volume, 1 ml, and this volume, 100 ml, 99% of the fluid is recovered. So these are the processes we have to talk about physiologically: filtration, reabsorption. The rest is excreted. Okay, so the kidneys are in the abdomen. They're higher than you think they are. They're actually tucked up under the ribs. So they're high in the posterior abdomen. We're going to have to get through the liver and the spleen and the pancreas and the stomach. We're going to see a lot of digestive tracts, organs, before we get to the kidneys. Kidney anatomy. This is going to be on the quiz. So this is a cartoon that shows some, but not all, of the names that I want you to be responsible, that you want to be putting in your head right now. You want to be aware that the surface, so let me go to the next slide, because the next slide has more of the detail. Okay? So the surface of the kidney has a name, and it's called the capsule. this slide actually calls it the fibrous capsule. I don't care about fibrous. The surface of the kidney is called the capsule. The outermost layer of the kidney, and we'll see this in the lab, is called the renal cortex. If you just tell me cortex, I'll be okay with it. But let's be clear that we have other cortices. We have an adrenal cortex, we've got a cerebral cortex, we've got a cerebellar cortex. We've got a lot of cortexes in our body. And today we're talking only about the renal cortex. So if you want to be, clear to, if you're talking to a physician and it's not clear you're talking about the kidney, you have to talk about the renal cortex. Okay? But if you just write cortex, where's the label here? If you just write cortex on a picture of the kidney, I'll accept it. The outermost layer is called the cortex. The middle layer is called the medulla. And again, we should officially call it the renal medulla because we have other medullas as well. But medulla is okay. So, capsule, cortex, medulla. The medulla has several structures within it. So the medulla represents this layer, and the structures within the medulla are the pyramid, and you can tell the pyramid because it's got these sort of stripes in it, and it has a name, but I'm not asking you to learn that name. The pyramid, the renal pyramid, and between the renal pyramids is a structure called the column, the renal column. And that's not on this slide, so you should annotate your PowerPoint by pointing to one of these spaces between the pyramids and call that a column. Pyramid, column, pyramid, column, pyramid. The tip of the pyramids is a little nipple, and so it's called the papilla, which is nipple in Latin. So this is the papilla of this pyramid. And this is the papilla of this pyramid. Pyramids in the medulla and papillas are the nipples that are innermost on each pyramid. We're going to be reinforcing this in the lab. The structure that the pyramids are connected to inside the medulla, so now we're getting deeper inside, these structures are called the minor calyces. So minor calyx, one calyx, two calyces. Just singular, plural. Wow. I won't take points off if you give me the plural instead of the singular or vice versa. Pyramid, capilla, minor calyx. Two minor calyces connected to a major calyx, spelling variant. And all of these major calyces connected to the renal pelvis. Again, if you just write pelvis, and we're looking at a picture of the kidney, I'm okay with it, but you know we have more than one pelvis. Well, now you know we have more than one pelvis. So you have your hip bone, but you also have your renal pelvis. The pelvis is connected to the ureter. The ureter is connected to the urinary bladder, which is not shown here. And the urinary bladder is what you control during micturition, urination. We're not going to talk too much about the control of micturition. This is, urine just drips out 1 mil per minute from the kidney to the into the ureter, and then just drains into the urinary bladder. Now, I should be clear, 1 mil per minute is what both kidneys produce. Each one produces about a half a mil per minute. So I think everything on this slide is fair game for an anatomy labeling exercise. This structure here, the tubes, the blood going in, the blood coming out, and the urine coming out, this structure here has a name, and it's called the hilum, the renal hilum, H-I-L-U-M. Those are the renal artery, the renal vein, and the ureter combined form the hilum. Between the minor calyces is a structure called the renal sinus. So each of these is a renal sinus, a space between the minor calyces. So minor calyx, sinus, column. So column is out here, sinus is in here. Questions? And so let's go back to the previous slide. I think I've covered everything that was on this slide. We're going to be talking about the nephron next. So here's a slide that's kind of blank, and you could use this to do a labeling exercise to reinforce your knowledge. Any questions about the anatomy?
You said the renal pyramid was the space between the medullas?
The renal pyramids are part of the medulla.
So what was the space in between?
The columns are the other part of the medulla. The outer layer is called the cortex. Did I answer your question? Yes.
Just to clarify, I sort of missed it. So each kidney produces 1 mil a minute or half a mil a minute?
Typically, our two kidneys combined generate 1 mil per minute of urine. If you have a kidney removed, your remaining kidney will produce 1 mil per minute of urine. It will adapt to produce more. But typically, when we have two kidneys, together they filter 100 mils per minute, and together they produce 1 mil per minute of urine. Okay, now we're going to get into the nephron. This is the functional unit of the kidney. So just like, a neuron is the functional unit of the brain, a nephron is the functional unit of the kidney, but it's multicellular, it's not one cell. So let's walk through this from a physiological and anatomical viewpoint. Here is so there's two nephrons that are being shown here. Here is one nephron, and here is another nephron. This nephron has a name, it's called a juxtamedullary nephron. It's this type of nephron that goes deep from the cortex into the medulla is a juxtamedullary nephron. So this is the cortex, this is the outer layer. Up there is the capsule somewhere. Cortex, medulla. This is a juxtamedullary nephron, and this is a cortical nephron, because it's mostly in the cortex. So let's talk about this juxtamedullary nephron. The differences are minor. A branch of the renal artery. Now, you want to make sure that you annotate branch, because this is not the renal artery. The renal artery is big. We can see it. Now we're getting microscopic. This is a small artery. It's a branch of the renal artery. It's not the renal artery. And coming off of this branch of the renal artery is an arteriole. And this is called the afferent arteriole. A as in afferent. So it's labeled up there. There is the word afferent. This is an afferent arteriole. This is an afferent arteriole. Afferent means going into. It's going into a structure called the glomerulus. So the afferent arteriole brings arterial blood into the glomerulus. And this is where filtration is going to happen. After filtration in the glomerulus, which we will talk more about in a minute, the blood leaves the glomerulus from the glomerulus it leaves in a structure called the efferent arteriole. So this is afferent to and efferent from. And I'm sorry they're so similar, but that's just the way it is. Afferent, blood to the glomerulus. Efferent takes blood from the glomerulus. It's still an arteriole, even though it has gone through a capillary bed. It's not a vein yet. That's one of the idiosyncrasies of this circulation. Blood from the efferent arteriole then goes either into a set of capillaries that are called, so this one is just not shown, but it's there as well. So blood could be coming out of here and perfusing this structure, which we haven't named yet. And these set of capillaries is called the peritubular capillaries. Peritubular capillaries, you have to imagine that they're here too. Peritubular capillaries. And now we have blood, arterial blood going, becoming deoxygenated, etc., and becoming venous blood. Here is another set of capillaries where the arterial blood goes deep into the medulla and then forms a capillary bed. And this structure is called the vasa recta. Vasa recta. So we have three capillaries: glomerular capillaries, peritubular capillaries, and vasa recta capillaries. And they each have different functions. Now let's talk about the tubules of the kidney. So surrounding the glomerulus is a structure called Bowman's capsule. And between the glomerular capillaries and Bowman's capsule is a space, thankfully called Bowman's space. So glomerular capillaries, Bowman's space, Bowman's capsule. This all forms the renal corpuscle. I don't think that name is on here, but this structure together is called a corpuscle. Are things okay?
Yeah, it's fine.
Leah? Yeah. Okay. So the renal corpuscle is comprised of Bowman's capsule, Bowman's space, and glomerular capillaries. Corpuscle. Probably should spell it. Sorry to keep moving around. Corpuscle. Question?
Is that called
Borman's corpuscle or renal corpuscle?
It's the renal corpuscle formed from Bowman's capsule, Bowman's space, and glomerular capillaries. Glomerular, derived from glomerulus. You get to say some good words in this part of the course. So filtration happens here, and then the filtrate collects in Bowman space, and then goes into this tube called the proximal tubule. Proximal because it is near the beginning. Some books, including the one that this came from, call it the convoluted tubule, because there are some parts of the tubule that are straight and some parts that are wiggly, wormy, convoluted. I am not going to make that distinction. So this is for me, this is the proximal tubule. But if you go on and become a nephrologist, you will learn the distinction. Nephrologist, a kidney specialist, you will learn the difference between the proximal convoluted tubule and the proximal straight tubule. For us, we don't need to worry about it. Proximal tubule. So now the fluid is moving through this proximal tubule, the fluid that was the filtrate, but it's now getting processed. Then it makes this deep loop, at least on a juxtaglomerular nephron, it makes this deep loop into the medulla, and this loop is called the loop of Henle. Loop of Henle. And it has a descending limb and an ascending limb. Your book sometimes, many books talk about a thick and a thin segment. And again, I am not going to draw that distinction. A descending limb of the loop of Henle and an ascending limb of the loop of Henle. And you're going to see that this structure, this loop-de-loop thing, is really important for water balance. It's going to be essential for making a concentrated versus a dilute urine. And if you're dehydrated, you want to make a concentrated urine so you don't become more dehydrated. So the vasa recta and the loop of Henle, they're not splayed out like this. This is a cartoon. They're all mixed together. This capillary bed is wrapped around this root structure, sort of the way this capillary bed is wrapped around this structure. You have to imagine. Okay, the fluid gets processed by the proximal tubule, by the descending and ascending loop of Henle, and then by the distal tubule. Distal because it's far away. I mean, it's actually right next to it. It's right next to it. But from a fluid flow point of view, the fluid went all the way around like this. So this is the distal tubule. The fluid leaves the distal tubule, enters the cortical collecting tubule, and then the medullary collecting tubule. Some books call it a duct, but it's not just a tube. It's also part of the processing system. So I call it the tubule, to make sure that the word represents that the cortical collecting tubule and the medullary collecting tubule process the fluid just like the proximal tubule and the distal tubule and the loop of Henle do. I mean, different details, but it's not just a duct. And coming out of the medullary collecting tubule, it goes into the renal papilla, which goes into the calyx, and it becomes urine. So all of those collecting tubules collect right here, and when the fluid that comes out then collects in this space. It's an empty space, or it's a urine-filled space. Minor calyx, major calyx, pelvis, ureter. So by the time we get to here, it's urine. By the time we get to here, it's urine. Here it is not urine. It's tubular fluid. Tubular fluid being processed. But by here, it's being processed, and it's urine. So we're going to be taking apart all of those details for the next 2 weeks, week and a half. Sophie?
So the nephron is in the pyramid?
The nephrons are in the cortex, the pyramid, and the column. So this shows a cartoon of how the nephron is situated in the kidney. This would be a cartoon of a juxtamedullary nephron. So you can see the loop of Henle going deep into the medulla, collecting tubule, dumping its contents into a renal calyx. Okay? So here is a flow chart of operation. I think I'll just skip through it and you can use this as a review, right? We start with filtration, then we go through the proximal tubule, the loop of Henle, the distal tubule, the collecting tubule, and then urine. Again, I am not going to differentiate between thick and thin, and I am not going to differentiate between convoluted and straight, but I am going to differentiate between descending and ascending loop of Henle. This is an important physiological difference. The other ones I'm going to gloss over. Okay, let's talk about filtration. Or other questions before I move on? Yes?
For the quiz, will we have the PowerPoint on the lungs that we didn't have on the exam?
Yes,
Okay,
that's on Moodle.
okay.
And I think I handed that out, but if I didn't give you a hard copy of it, I will. But it's on Moodle. You're talking about the respiratory one that,
Yeah, like I'm talking about the quiz. Will that material be covered on the quiz?
oh no, it will not be on the quiz.
Okay,
It'll
I
be
see.
on the exam, but
I
not on the
see.
quiz.
Okay, okay. So the quiz is just what we do today? And the
Today
quiz.
and, yes, today, period.
Okay.
And lab. Today and lab. Okay. So this is filtration. Now we're going to talk about the molecular mechanisms of filtration. And it's really anatomical. So this is a blow-up of the renal corpuscle. Right, the renal corpuscle. Bowman's capsule, Bowman's space. Glomerular capillaries. Afferent arterial, efferent arterial. This is showing the fact that the distal tubule makes a contact point with the afferent arterial. We'll talk about, this is going to be important. We'll talk about this on Monday. The distal tubule makes contact with the afferent arterial. Just to be clear how that's possible, the distal tubule making contact with the afferent arterial. Right there. Okay. Bowman's capsule, Bowman's space, renal, the glomerulus, afferent arterial, efferent arterial. Blood comes in the afferent arterial and is filtered by these glomerular capillaries. The glomerular capillaries are specialized for being leaky. They're specialized for allowing filtration. They're specialized for being leaky to fluid. That's their job. And so these glomerular capillaries are surrounded by a group of cells called podocytes. And podocytes have these interdigitations, but with spaces in between them that are called filtration slits. So the podocytes wrapping around a glomerular capillary do not form a tight seal. the endothelial cells are leaky and the podocytes are leaky. So that fluid can leak out of the glomerular capillaries into Bowman space. That's the filtrate that the kidneys make. So this cartoon is actually a pretty good one, because if you look, the next slide shows actual data from an electron micrograph of glomerular capillaries. So in this case, the Bowman's capsules have been removed. So we're seeing glomerular capillaries. Here is an afferent arteriole, and here's an efferent arteriole. Here's a branch of the renal artery. I don't know what ILU stands for. Branch of the renal artery making several afferent arterioles, one of them going to each glomerulus.
Oh,
Yes?
the filtration slips in the podocytes, are podocytes like the loops and then the filtration is in
The
between?
podocytes are the cells that wrap around the glomerular capillaries, and so this is one podocyte, this is another podocyte, this is the nucleus of a podocyte, this is the nucleus of the other podocyte, and they connect to each other through these leaky junctions called filtration slits.
Thank you. You said it leaks into Bowman space?
It leaks, the fluid leaks into Bowman space. So the fluid leaks from the plasma going through the glomerular capillaries, through the endothelial cells, and we'll talk about the windows, the fenestrae in the endothelial cells. So holes in the endothelial cells, slits in the cells that surround it, support it, and the fluid connects in this space here, and it's going to go out the proximal tubule. We saw this one. Here shows you that cartoon is actually pretty good. Here is the cell body of a podocyte, and here are those connections, the filtration slits. So cell-cell interdigitation, but with spaces between them, wrapping the endothelium of the glomerular capillaries. Let's skip this one. This is a high resolution, a high amplification micrograph that just shows the capillary having holes in it, and the cell body of the podocyte having slits in it. So lots of ways for fluid to move. Here are the holes. So this is from the inside of a glomerular capillary. So we are inside a capillary, and if we are a water molecule inside the glomerular capillary, we have a chance of being squeezed out through this hole, and then coming out through a slit, and then entering Bowman space. It's not often that I get to show data that really makes the cartoons look real. That's why I'm doing it. So, filtration. Now we're going to get into physiology, not anatomy. Let's get oriented. This is let's just say this is an afferent arteriole, glomerular capillary, efferent arteriole, Bowman space. Okay, so this is now we're not talking anatomy, this is highly simplified. Afferent arteriole, blood coming in, filtration going to happen here in the glomerular capillaries, and then the efferent arteriole where the blood is going to go into another, either the peritubular capillaries or the vasorectal capillary. What causes filtration? So I want to be clear. What allows filtration are the holes in the endothelium and the filtration slits. Those allow water to flow. But what forces water to flow is what we're talking about now. What the fenestrae, the windows, the holes on the inside of the endothelium, the filtration slits between the podocytes, those allow water to flow, but what forces water to flow is pressure. So pressure in the glomerular capillaries pushes water and whatever the water can carry into Bowman space. That's what this arrow is. Pressure, hydrostatic pressure, blood pressure in the glomerular capillaries pushes water into Bowman space. There is some pressure in Bowman space that by itself would push water from Bowman space into the glomerular capillaries, but the blood pressure is almost always greater than the pressure in Bowman space. So this minus this is the pressure differential. So we have glomerular capillary pressure favoring filtration, and we have Bowman space pressure opposing filtration. In addition, we have osmotic pressure. Now we have to understand osmosis. Some of you may have studied this in chemistry. Osmosis is the movement of water in response to a difference in solute concentration. So if we have a membrane that is permeable, that's what the hole illustrates here, and we have a high concentration of sodium chloride, some solute, on one side, and we've got a low concentration of sodium chloride on the other side. Now this is not a channel, this is a hole that allows water to flow. Which way is water going to flow in this scenario that I have illustrated? There's no difference in hydrostatic pressure, but we have a difference in solute concentration on the two sides of these holes. Which way is water going to flow? From your left to right, or from your right to left?
Right to left.
Right to left. Water is going to flow from the low concentration into the high concentration. Water. This is water flow. That's osmosis. That is osmotic pressure, forcing that flow of water. So now you can see that the osmotic pressure in the glomerular capillaries is lower than the osmotic pressure. So the pressure differential for osmosis is in this direction. That means that there must be higher concentration of solute here compared to here. Higher solute, lower solute, osmotic pressure. Okay, so we're almost there. What I have to tell you now is that when fluid flows through these holes, small molecules flow. Salt, sodium, chloride, potassium chloride, phosphate, calcium, all the goodies in terms of the small molecular, all the ions that we talk about, they are small enough to fit through these holes. That's key. Water and small molecules. Cells, way too big, typically. Proteins, way too big. Glucose, small enough. So glucose in the blood glucose, you know that you have serum glucose. Blood glucose flows when water flows. So water, all the salts that we talk about, glucose, other small molecules like ATP, but not protein and not cells. Nutrition.
So when we do a urine test and there is a concentration of red blood cells and proteins, that means that your kidneys are damaged, right? Or what?
If you see red blood cells in the urine, that
Or
suggests
protein.
glomerular damage.
Yeah,
If you
okay.
see protein in the urine, that sometimes that's normal and sometimes it's not normal. So for example, after you exercise vigorously, it is not uncommon to find some protein in the urine.
But how
that's
do
not-
they fit? How do they fit through those holes?
There is sort of, so proteins, especially albumin, are just at the limit
I see.
of between small and not small.
I see, okay.
So higher pressure, a little bit of, force, and you can squeeze some albumin through these holes.
I see.
But a lot of albumin in the urine is indicating kidney disease. So what forms this osmotic differential is not salt, because salt is freely filtered. It's protein. The osmotic force of protein in the plasma is what makes the solute that makes this arrow is mostly protein. And most of that protein is called serum albumin. In cell-phys, you worked with bovine serum albumin. That was your standard protein in lab, remember? BSA, bovine serum albumin. Well, bovine just means cow. So you have human serum albumin, HSA. And a cat has feline serum albumin, and a pig has porcine serum albumin. You worked with a cow variant, bovine. But we have albumin in our blood that gives rise to this osmotic pressure. Okay. Sorry. So now, I think you have enough material to work through the first set of clicker questions and then some labeling exercises. So on the last 2 pages of the handout, and I'm sorry I made them one single-sided for a mistake, I want you to work individually or together, but be safe, on the clicker question, this one, and the labeling questions. Let's do this for 10 or 15 minutes. We'll take a break, we'll shut down the recording, I'll answer questions. But the act of writing this out, I know you haven't memorized it yet, but the act of looking it up and writing it out, that's part of the memorization process. So you're going to do that today, now, for the next 10 minutes or so. You'll do it in lab, we'll have an exercise in lab, and then you'll have to show me that you put it in your heads on the quiz.